Hello, my name is Adil Yusuf, and this is our project Increased Risk of Psoriasis, along with my partners Ashley Kubanon and Hannah McMills. So the goal of this project was to be able to use an ancestry dataset in order to determine traits about an anonymous donor or their risk of developing a certain disease. In this case, we picked psoriasis. And some other goals of the project were to be able to understand SNPs or SNPs and how they can be associated with diseases. So SNPs are actually a change in a single nucleotide of a gene. Oftentimes in a regulatory region, this is going to cause a change in the gene's function. And SNPs are actually identified with RSIDs, which label specific SNPs. These RSIDs are going to be associated with certain diseases. And the SNP that is associated with psoriasis is RS126551A1. And over here in figure 1, we can see an example of a SNP where one of the nucleotides was changed out, making a single nucleotide polymorphism. So what is psoriasis? Psoriasis is a chronic skin disorder where oftentimes you're going to get flare-ups of symptoms. It actually occurs commonly in younger ages, and you're going to be getting skin lesions, flaky flakes, and scales that are going to be hurting and itching on your skin. And over here in figure 2, here's a showcase of some of the symptoms. So this slide sums up some of the associations that we found with psoriasis, but most importantly, we used the OMIN database to find an entry about psoriasis that included an SNP on there, and then we used SNPedia to search up the SNP that we found from the OMIN database. This SNP ID was RS126551A1, which is associated with the HLA-CW6 allele. So once we found this RS number on SNPedia, we acquired two datasets 1 and 2. These had genotypical data from 2 anonymous donors, and companies that provided this information will also be anonymous for privacy purposes. So we searched this RS ID number on the datasets, and we found it in dataset 2. Thus, we're going to be using anonymous donor 2 and predicting their traits based on this SNP.
So moving on to results and interpretations, we see from dataset 2 that our anonymous donor has a genotype of GG. Furthermore, we also see in the bottom photo that our anonymous donor also has an X and a Y chromosome that's present. And so this suggests that our donor is male. And to kind of interpret what a genotype of GG means for our particular donor is that we see on this table that a genotype of GG suggests that this individual has a possibly increased risk of psoriasis. And to continue off that, this kind of figure shows the 3 different genotypes that we saw in the previous slides depicted by the colors orange, green, and blue, and what percentage of a particular population has this genotype for this SNP. And so we see that a genotype of GG, which is designated by the color blue, is only found in one population, which is the CEU population. And so the CEU population refers to the population of Utah residents of Northern and Western European ancestry. So to conclude, what we found in our data is that we predicted that the donor from dataset 2 is male and a Utah resident of Northern and Western European ancestry that has a possible increased risk of developing psoriasis. And so to kind of continue off what we learned about this research is that, we understand that potentially anyone could analyze and interpret their own DNA test results and know what type of risks and diseases and traits that they may have or their families and friends may have, and how that can affect them and their health. And so this was a very valuable experience for us to be able to see the process of how geneticists may interpret data like this. And so to conclude, we would like to thank Dr. Joseph Ross and Sose Kendavian for guiding us and providing us the resources that we need to complete this project. And lastly, we would like to thank the audience for taking the time to listen to our presentation. Thank you.